Good morning or good afternoon, ladies and gentlemen, depending on where in the world you're joining us from, and thank you for joining today's webinar. Today's webinar is brought to you by the Business Review and is in conjunction with Freeslate. Today's presentation will be applying high-throughput research to biologics pre-formulation. Today's presenter will be Eric Carlson, who is the Vice President of Collaboration and Solutions Development at Freeslate. Before I hand over to Eric, I just want to run through a couple of items that will be of importance to you today. On the right-hand side, you will notice there are various functions that you can use in this session. For the purposes of today's presentation and the large number of people that are online, we will not be using the hand raise function. We will not be making any of the attendees' lines live. However, if you wish to communicate with the presenters throughout the session, please submit your questions via the question box that is available. What I would suggest is throughout the session, submit the questions as soon as you think of them. The reason for this is we will prioritize them in the background and we will answer as many of those questions in the final 15 minutes of today's session. Midway through today's presentation, we will be having two polling questions. We ask you to participate in the session and submit the appropriate answer when the time is due. I will take over at that point and actually help you through that part of the session. Okay, well, without further delay, Eric, if you're ready, you now have control, and the presentation is yours.
Thank you very much, Kane, and thank you everybody for attending today. So today I'll be talking about high-throughput research for biologics pre-formulation. I'll be spending some time giving a broad overview of Freeslate approach to this topic and some general case studies. What I thought I'd do is start off talk a little about who Freeslate is. Some of you may know Freeslate, and some of you may not. We are a heritage. Our heritage is a spin-out from Symix Technology. Symix was a pioneer in high-throughput research for materials science, materials development for over 16 years, and had quite a long list of intellectual property and high-throughput approaches and microscale technology over that time period. Freeslate spun out about 1 year ago, and since that time, we have about 100 employees. We're located in Silicon Valley in Sunnyvale, California, and we serve a broad array of sectors from the life science, pharma and biopharma, as well as home and personal care, and a number of areas of catalysis research. Today, in particular, I'll be talking about biopharma and looking at pre-formulations of biologics therapeutics. So really, what Freeslate does is we really are focused on high-throughput research and high-throughput research solutions. When we talk about those, they're really comprised of three things. We do provide proprietary automation platforms, and I'll talk about some of those later on and talk about those relative to our case studies. In addition to the automation platforms, we have a set of informatics that really ties the entire workflow together. It's called the Laboratory Execution and Analysis Software. It's a suite of software designed to seamlessly integrate from design to analysis, and it can even be used to tie together researchers across multiple sites. Finally, we have a pretty deep base of scientific expertise and can provide collaboration. Here in Sunnyvale, we have a set of labs with a set of application scientists and engineers to really work together to continue to develop applications and to work with our customers, scientists, to develop work for them as well. So we're going to be talking a bit today about biologics, and in particular, as a little bit of background, when we think about where high-throughput research can have an impact, we're mostly talking about development. Historically, high-throughput approaches and combinatorial approaches have been used extensively in drug discovery space very early in the process. Generally speaking, where we have found a real impact and a real need that's been underserved is in the area of development. And this is an area where you're trying to determine developability of a compound and really try to, there's a need to establish a wider design space. These are samples that tend to be more complex when you're making the samples. These are, and these also tend to have much more complex data sets where you're bringing in data from multiple different types of instruments and trying to bring them together. And so systems and approaches that can enable one to explore a broader formulation space or pre-formulation space and tie that data together can become very impactful. So generally speaking, the areas that we'd be talking about are the area of pre-formulation development. We're talking about solubility, stress testing, early formulation development. Formulation development, where we're looking at excipient studies, stability, looking at tolerance, tolerance and robustness, and process development become very key. specifically today, we're going to talk about biologics. And along the lines of what I just talked about, this is an area where the formulations can be quite complex, and I would suspect that most of you on the line today are familiar with the complexity of this particular space. There's quite a broad inherent factor space that one can explore, from looking at the composition space, looking at a variety of different pH, salt levels, excipients, and different concentrations when exploring different potential formulations. There's also a variety of different environmental factors. You can look at process operations, handling, potentially shipping and storage stresses, temperature, light types of stresses, agitation and shear, surface exposure, freeze-thaw. All these things can contribute to the stability or the lack of stability of your formulation, and it could be important to explore the effects of these on the early formulation development. And finally, of course, there's a variety of product factors from dosage forms, what form the container closure is, shelf life, and the route of administration. Really, and the other factor with this, in addition to all the complexity of the formulation space, is that there is not one single type of analytic that one can use when trying to assess your biologic formulation. Typically, a researcher would use orthogonal analytics to assess multiple different types of biophysical characteristics. And that means that there's additional complexity, because now you have a number of samples. Each sample you may want to assess with a number of different analytics. And so if you're going to apply a high-throughput research, you need a strategy that can take into both those things into account. So generally speaking, when we're talking about implementing high-throughput approach to pre-formulation activities, there's a number of different objectives that one wants to achieve. You'd like to be able to explore a comprehensive biophysical characterization of formulations and be able to use orthogonal analytics to do that. Really, this is a space where, as most of you know, early in development, you've got very limited amounts of material, and material is very precious. So the ability to screen additional formulations or screen more broadly with smaller amounts of material becomes extremely valuable. So going to smaller scale and being able to screen broader can be very impactful. And really like to perform those things without adding additional time or resources. As much as we all would like to be able to potentially just take much more time or maybe add more people to the team and more staff, the realities of the business world today don't really allow for that. You're really trying to do more with less or more with the same resources that we have today. And finally, most organizations are working on campaigns or series of different types of formulations, and so the ability to build organizational knowledge and be able to tie things together and be able to leverage past experience becomes very important. So relative to that, the types of requirements that one would want to have is, first thing is you really need to be able to have an integrated control of diverse types of analytical devices. And that means these are devices coming from multiple vendors. They typically have multiple types of analytical control. The data types and the data models look different. And so, but you need a way to be able to control the instruments, be able to capture that data, and be able to display and report that data out in a common way. Secondly, you need to be able to handle complex, low-volume sample handling and be able to do measurements on those small amounts of samples if you really want to achieve high throughput. This is an area that I think many of you know is not totally completely developed, where people are still continuing to add and look at new techniques and new approaches, which means really one wants to have a modular approach. There could be, I think we all know, there could be new analytics coming out in the next year or 3. And so one wants to be able to take advantage of any improvements to new analytics, improvements to new approaches. And so a true winning approach will be modular and will have the ability to swap in and change over time. And finally, you're going to be generating a lot of data, so you want to be able to handle the management and reporting of rich data sets from a variety of sources. So with all that as a background, knowing that is the goal, I'm going to talk a little bit next about Freeslate's platforms and our underlying platforms to achieve that, and then I'm going to talk a little about some of our case studies as well. So the first platform I want to talk about is some of our informatics. So I mentioned that we're going to be talking about automation in a bit, and automation and robotics is a logical place to think about with high throughput. But the other one that's equally as important, perhaps more so, is a system to really handle the informatics and the data that's coming through from all these different systems. This slide describes an informatics platform to empower decision-making for high-throughput types of research. Here along the left side, there's a series of arrows that talk about from design, execute, analyze, and report. The idea here is that this is generally the process that a scientist or researcher would follow when they're going through any of the work they're trying to do. You need to design sets of experiments, you go into the lab, you execute and you prepare samples, you need to process them, you make your analytical samples, and you need to analyze them, and then you need to report out and visualize the data and share that with your group. These are activities that happen in different locations. The arrows here in white are arrows that actually typically happen at a researcher's desk. And so what we've done is we've actually set up a suite of software. This is our laboratory execution and analysis suite. And there's different programs that approach different parts of this process. The design is handled by a program called Library Studio. It really enables array-based experimental design. And it's a very intuitive program for researchers to use. I often describe it as an Excel for chemists. When you're in the lab, then, you need to be actually making samples, preparing, processing, and then analyzing samples. And these are all things where you're either a researcher or typically equipment is going to be hands-on with the samples themselves. And so here we have a set of software called Automation Studio. It really controls the various pieces of robotics I'm going to talk about. It's also the same software that actually wraps around a series of analytical instruments, allowing one to control them, to capture the data from them, and be able to take the data and store it all into a common database. Finally, when a researcher is back at their desk, they need to be able to report things out, be able to visualize data, do some data processing, and generate reports. This, again, happens at the researcher's desk. And there's a set of software called PolyView that really enables enterprise access to the raw and the summary data for visualization. One of the advantages of this type of approach where things are separated out from a design, an execution, and reporting scheme is it really supports the ability of doing globally dispersed types of collaborations. So really, you could actually have a researcher in one geographic location doing designs and designing sets of experiments. You can have a lab set up in another geographic location where the execution is actually occurring, and they're pulling up designs and recipes generated by the first researcher and executing them in the second lab, and being able to then capture data either within the lab or in a lab down the hall, and all the data be going to the same database. And researchers then in a third location or back at the original location could then view all that data through the database, be able to pull that up and report that out. And so as the slide says, it really does enable global collaboration and data sharing by integrating all this data into a single workflow. And it goes to really building organizational knowledge. So in addition to talking about the informatics that's there, we have a number of, we have a robotics platform that we also use. There are a number of different robotics platforms that are out in the market. I would imagine that many of you online are familiar with a number of the different types of liquid handling robots and things that are out there. The core module that Freeslate has is a little bit different. It's designed to handle things beyond simple liquid samples. First of all, it's designed to handle fairly complex samples where it can dispense viscous liquids, solids, powders, flakes. It can do aqueous, organics. It can do heated liquids, a whole variety of different materials. So it really can dispense and prepare a wide variety of different types of samples. On the deck, it can do things like viscous blending. It can do heating, cooling, vortexing, different types of mixing. You can connect up to centrifuges to spin things down, a variety of different processing types of characteristics. And then there is some on-deck types of analysis that can be done as well. There's a on-deck integrated balance, so you can track everything by mass. There's an integrated camera, so you can image samples and do image analysis and be able to do visual inspection in an automated way. There's a pH measurement, so you can track the pH of your samples and create samples of various pH. And in addition, you can integrate with a variety of third-party instruments as well. It's a very modular system, and so these are all set up with each of these unit of operations being an individual functional element on the deck that either sits on the robotic deck itself or sits on one of the various functional robotic arms. And the robot is typically configured to be, you have the right set of unit operations to be appropriate for your set of chemistry. The other nice thing about this is that because of the modularity that it has, it's a system that can actually change over time, so that if a new analytic came available, or there was a need to change in the way the protocol was needed to be run, it'd be fairly easy to either change the protocol or to swap out some of the unit operations to take advantage of that and be able to modify your protocol going forward. I mentioned about the integration of instruments, and typically for protein preformulation, there's a number of potential instruments that one may be interested in. There are things that have to do with processing, where you may want to integrate with a centrifuge to be able to spin down samples, to be able to separate samples. There are incubators and different types of samples to provide either incubation or different types of thermal or environmental stressing of the samples. Various types of plate sealers. The on-deck robot, on deck in the robot, we actually have a capping and decapping to cap and decap individual vials. Oftentimes, you may want to actually seal individual analytical plates to avoid evaporation. Dynamic light scattering, UV vis, and fluorescence are very typical measurements. There's a variety of HPLC measurements, of course, from SEC to reverse phase to ion chromatography and DSC. These are all very typical types of analysis. And really, I think that for a researcher to do the job they want to do, they don't necessarily want to choose or rely on a single one, but they really want to be able to capture the data they need from any of these. There could even be things to look at particle assessment, things like MFI, CIEF, or CSDS as well. So there's a couple of different ways that these can be integrated into the system. A typical way, integration can happen in a couple of different forms. One way of integration is a virtual integration, where this robot itself sits on a bench. The other analytics sit either on a bench across the room or another place, and so they are physically separated from the automation station that's preparing samples, but they're tied together through the software, so that a sample that's prepared on the core module robotic deck would have a sample array. There'd be a barcode associated with that with an identification number. A researcher, when it was done, would carry that array of samples across to a particular analytic, scan in the barcode, and then from that, the software at the analytical station would know how that sample and that set of samples relates back to the way it was designed, and be able to store that data back to the database. In other ways, you may want to have a more physical integration, and there's a number of different modes of physical integration where the robot itself is directly connected with the various analytical pieces and can hand samples off directly. I think my next slide just talks about some of those different modes. So, and really, we've done a number of different offerings from a fairly standalone type of mode, which relies almost solely on virtual integration, to a fully automated mode. And it very much depends on the organization, the throughput, and what type of workflow one is trying to enable. So the different modes I'm showing on this slide, the first is a benchtop mode, where you have a standalone core module designed for really setting up, preparing samples, and being able to prepare those samples alone. Samples then, as I mentioned before, could be carried off to the variety of analytics that may be available to be used. A second choice would be a standard core module 3. This particular generation of the instrument is called a CM3. That may have one or two physically integrated instruments. The CM3 itself, this version of the core module, is typically a 1.5 meter robot. It has the ability of having, and the option of having a 2 meter robotic rail, so the rail itself can overhang a portion of the deck. That overhang could actually overhang analytical instruments. And so you could actually use the robot to prep samples on the deck, and have the robot pick up and take samples and pass them off to some number of analytics just beside the robot. Other analytics could then be handled in a virtual integration mode. Option 3 would be a highly integrated mode, where you would either use the extended CM3 footprint with 5 or more analytics, and as shown here, you actually may be tying 2 of the CM3s together, one to do sample prep, as shown here on the left in a very small picture, and on the right, this is the CM3 that's really serving a variety of analytics, and all it's doing is shuttling samples between the various analytics, between that and a carousel for storage. And finally, there's a mode of doing a fully automated mode, where this is now a very large, essentially you've got a setup here that would fill an entire laboratory. You've got a large, I'm showing a large U-shaped table and a U-shaped configuration. We've got 2 core modules set up on a corner of the SIS setup. Down the middle of the U is a 6-axis articulated robot that's able to shuttle samples in a hands-free mode from environmental chambers in the back corner to these sample prep robots to a variety of HPLCs, SEC, reverse phase chromatography, ion chromatography, or other types of analytics, whether it's DLS, CIEF, or a variety of different types of instruments as well. So we've actually have experience in doing all these different modes, and different ones are appropriate for either different throughput needs, different capacity needs, and different types of workflows entirely. Particularly for the biologics development, one of the modes that we're actually really very focused on is this highly integrated mode. And our target really is to support pre-formulation screening and stability workflows with a combination of vials and microplates. It does have a modular design, so it can substitute in a variety of robotic elements and analytical tools. And as I'm showing here, there's again a core module based for looking at analytical prep, and then a core module for serving the analytical needs. This core module is focused on formulation preparation, sample analytical prep. It has an integrated pH and visual inspection station, as well as capping and decapping and a variety of different types of dispense technology. It's joined in the middle by a carousel that can either have additional storage capacity for different types of consumables or to store up and prep various analytical plates that can be queued up for the analytical that's going to happen subsequently. The second core module is really additional, where the deck itself is only comprised of a variety of analytical and perhaps things like a centrifuge and different types of processing equipment, where the robot would pass off to a second robot, and this robot would just be busy serving up a variety of different types of prep and analytical pieces as well. As shown in the schematic, the integrated components are a dynamic light scattering instrument, a UV-Vis fluorescence plate reader, centrifuge, a plate sealer, a shaking integrator to allow you to basically thermally expose the material at a given shaking condition and agitation condition. It also has a pH and a viscosity measurement for doing early assessment of formulations. And the entire thing, as shown, is done in a HEPA enclosure to provide a low bioburden workspace as well. So I think with that, actually, before I move on and talk about some of the specific case studies, I think we're going to pause briefly. I'm going to pass control back to our moderator to let him provide and do some of the polling.
Fantastic. Okay, thank you very much, Eric. Okay, ladies and gentlemen, I'm now going to launch a poll for you. You can see the question on your screen at the moment. You will be given the 5 options that are displayed. Please select the most appropriate option for you and click the Submit button at the bottom. You'll have around about 30 seconds to do this. Eric, do you want to explain to the audience why we're carrying out this first poll before we move on to the second? Okay, we've got around about 15 more seconds left for you to complete the poll, ladies and gentlemen. You'll see the results come up on your screen in just a moment there, Eric. Can you see them coming up on the right-hand side there for you?
I am looking. Yes, I do.
Fantastic. Well, they'll be launched to the audience in just a moment. So we've got kind of just over 75% of the audience have now completed it. We've had it open for about a minute now. So let's have a look at the results. Okay. And you should now be able to see the results. So if I hand back to you here, Eric, if you want to just kind of give your opinion on the results.
Absolutely. I think this is it's not overly surprising. I think HPLC is a real workhorse across a number of different applications. I think I'd be surprised if I didn't see HPLC lists up there, and there's a variety of different liquid chromatography methods that I'm sure if we dug in and asked more questions that would be probably underlie that. I do think that I would expect to see people using dynamic light scattering and UV-Vis as well. And the MFI and Hive are ones we'd asked about because these are ones that I think are becoming more common, and as people try to assess the appearance of particles and aggregation and the stability of things as well. Fantastic.
Fantastic. Okay, should we have a look at the second poll now?
That sounds great.
Fantastic. Okay. Ladies and gentlemen, you should now see a second poll on your screen. You have two choices here, just simply yes or no. If you could just quickly complete this one as well, click submit. I have around about 30 seconds to deliberate. Okay, it looks like most of the audience has now answered. We're just shy of the minute mark here that's been open. So should we have a look at the results, Eric?
I think we definitely should.
Fantastic.
Excellent. So I think what the results show is that that's 80%. That's 4 out of 5 people that answered the poll, and it looks like actually we had a pretty good response rate overall. Are interested in viscosity. I think that's actually interesting. We'll talk about that in a bit. This is one in particular that there historically haven't been good methods to look at viscosity on small amounts of sample, and I think it's not a traditional one that's used particularly in early development. And so our feeling has been there's a need here, and we'll talk a little about that later on here as we go through, after we go through the case studies. Great. So shall we proceed?
You now have control.
Okay. I have a couple of case studies that we're going to talk through just to show some examples. And so the first case study, actually go back, is a case study on looking at solubility screening and doing that in high throughput. The reference is down below. This is really the ability of using PEG, polyethylene glycol precipitation, to facilitate high throughput solubility screening. This is a methodology that's been developed and has been known about for quite some time. And the idea is that by adding small amounts of PEG, the polymer itself will decrease protein solubility. It does this via an excluded volume mechanism, and it basically pushes the proteins out of solution. And the amount of PEG needed to precipitate protein is correlated with the relative solubility of the protein in whatever buffer you're working in. So quantitatively, one can do this and look at the appearance. The solubility can be estimated by extrapolating back to zero, the zero concentration of polymer. You can also do a qualitative ranking, looking at relative amounts of PEG needed to induce protein precipitation, depending on the speed and how you're doing things. So generally speaking, you can do your design and do a design of experiments or do an array-based design to explore the conditions of what one is interested in. One could do an array-based dispense to create a variety of materials, do your assay assembly, spin things down in centrifuge, and in this particular case, the assessment is all done with an absorbance measurement using a plate reader. In the blue box, it shows a little bit more detail about this workflow in particular. We would combine the buffer and combinations of 40% of this PEG solution to get the desired PEG solutions. and they'll mix these thoroughly. We then add those solutions to a plate, add the protein on top of that plate as well, and then centrifuge it after 24 hours. Transfer equal volumes to a clean plate and measure. So you can do this, very quickly and build up curves. So this slide shows a comparison of automated and manual approaches. And it's not surprising. This is, we're not really changing the underlying science behind it. The automated approach shown in the red overlays the manual approach shown in blue quite well. And what we're showing here is a concentration of an antibody at various concentrations of this PEG 6000. And what you can see is the concentration falls off dramatically at a certain concentration of PEG. One of the things that allows you to do is that you not only do you get comparable results, but because you get comparable results, one can do this much more quickly, and you can do a much more thorough exploration than you probably would do manually. And really what you can do is you can more thoroughly explore this region that's circled here is this transition region. And it really allows you to more thoroughly explore that region and understand, okay, what actually is that particular region, and you can get additional conditions and additional data there that really allows you to pick out more precisely where the transition and inflection point could be. So the approach that we took was really a qualitative approach to really look for speed and look for relative solubility. We were looking at rank ordering based on approximate % PEG transition from a soluble to insoluble protein using a curve fitting algorithm to fit those sigmoidal types of curves. There aren't really disadvantages. We didn't need a quantitative approach in this particular stage, but there are some large advantages. One is you don't have, there are some difficulties in doing a quantitative extrapolation. If I go back here, extrapolating the sigmoidal curve back to 0%, there's a fair bit of error involved in trying to extrapolate this shaped curve back to zero concentration. And being able to pick out the transition is much easier, much more precise way of picking this out. You can generalize the PEG range and increments, so you don't need to do multiple experiments to focus on the equilibrium range. And we can do a number of different conditions per plate to really explore a broad range of conditions. And so really, I'm showing some representative data, but one was able to see with different types of buffers, from citrate, acetate, histidine, and phosphate buffers, a number of, this is showing the solubility at the midpoint. And what you can see is, as a function of pH, the solubility of this antibody changes quite a bit and allows you to quickly map out the relative solubility in different types of buffers very quickly. A couple other case studies on some array-based formulation optimization. These two, so again, we're trying to apply array-based formulation to evaluate, again, as we're looking at solubility of and using a different approach. And we'll compare these automated approaches with a manual approach. Here we're using the robots to actually weigh out and we're doing this by mass, so you can actually weigh the automation formulation vial. You can aspirate the surfactant from a source into the vial, dispense the surfactant, and weigh the formulation and surfactant into that to know exactly how much is there. And then when you do your assessment, you can get an idea about exactly how much, you know the nominal concentration, you can calculate the actual concentration as well. A little more details about the workflow itself. We do the design in this array-based software. In this particular case, we developed a bulk lyo formulation, and we performed the lyo directly into a plate. The robot then prepared a master plate of formulations based on the design. We transfer these master solutions into the lyo samples using the robot. We equilibrated for a period of time. We then measured the pH of each sample and imaged each sample. We then did a filtration. We did and we performed reverse stage HPLC on plates that we diluted out, analyzed for concentration and purity. And this was done and captured the data. So the design we're looking at was a fairly broad space. You're looking at here at these were done here in a 96 well plate. On the left half of the plate, there was no surfactant. In the right half of the plate, it was a small amount of surfactant. From the top to the bottom of the plate, we were changing the tonicity modifier. And then going from left to right, we're changing the pH. So you're seeing essentially 96 different conditions as we go across the plate. So this slide here shows some results of what the results of doing a high throughput approach looks like. The schematic on the left shows the solubility detail coming through. These were all done at under 500 microliters of sample, so it's a very small amount of sample. We actually are, what you can actually see quite quickly is it really maps out the solubility profile. And you can see quite quickly where the solubility falls off and how it falls off as a function of pH and how it falls off as a function of tonicity modifier across there. On the right-hand side, it compares the manual, this high throughput approach with a manual study. Again, the data is very comparable, but it's not surprising that by preparing all the samples at the same time, by making all the samples, not only is the data very comparable to each other, but it takes much less time. You can actually complete this type of study and only from start of doing the LIO to finish of getting the data, only in a matter of days, whereas doing it manually, researchers probably do it over a series of days or a couple of weeks to actually get all the data and do everything. If one was using a manual approach, you would unlikely do all 96 conditions, and you wouldn't have this type of granularity as well. So you can really look at rank order and look at solubility with much less time and much less material and be able to explore a much broader space. This particular example, this talks a little about the another surfactant composition formulation space, where again we're looking at different types of surfactants and solubilizers from going across the from solubilizer 1, 2, and 3, and then different combinations of them going across, and then different amounts of total surfactants going from top to bottom, and with the first column being controls using a particular protein. I'm showing here an example of the visual inspection and visual identification of precipitation. I mentioned about the fact that we have an on-deck camera. I'll show a little bit more about that again later. One of the things when somebody does things manually is that you have you can see all your samples. And researchers are very used to being able to look at the samples and be able to see what happens. And if you're shifting to a mode where you need to do many samples at once and potentially hundreds of samples, it becomes very difficult to actually look at and visualize and stay as visually connected with each sample. So by providing an on-deck camera, it's a way to actually maintain that and be able to see what's actually happening in the samples. So it's a side view image. These are 2 mil, essentially LC automation vials. And what we're seeing here is a control that just has no precipitation and one that does have precipitation, and you can actually pick that out. And the robot can pick that out using, these are all taken under constant light conditions, and you can pick that type of thing out with a image analysis as well. The other thing is it stores a record of the picture to your to the database and whatever electronic record keeping you actually have, so that if anything ever happens funny with a sample down the stream, you have a record you can go back and you can see, was there something funny about the sample? Did it change a funny color? Was there something else about that sample that I'd like to look at? In addition, these samples you can also do dynamic light scattering and SEC, looking for the additional appearance of particulates. Here too, what we can see is that here where you had no surfactants, you're going again from A through F with no surfactant, different amounts of surfactants moving from top in this display to the bottom. You're seeing increasing amounts of surfactants across here. You can see we're seeing less and less precipitate going across. One thing is the image analysis and then the DLS as well is a very fast method, whereas SEC is slower. And I noticed that some 70% of you all do rely on HPLC, which is a very robust method, and it's the data that we all know and trust. It's slower, and so if there's a way to complement that technique with the techniques that are faster, you can get a large advantage. In this case, you get very similar data, and you can pick out using your image analysis and your DLS where you're seeing particulates, aggregates, and so you can feed that and use that to choose which samples actually go to your SEC. And on my next slide, I show a screenshot looking at the chromatograms coming from the SEC. What one can see is that there are blanks here, and the blanks aren't a mistake, and they aren't data that we accidentally missed. They're data that we intentionally excluded, and actually the robot intentionally excluded. It knows, based on the image data and DLS data, these are samples that have already crashed out, and they're already precipitated, and so it doesn't need to actually take the data on those wells. It can be avoided, and so you can be more efficient with your SEC time, and you can be more gentle on your columns as well. Great. So I'm going to change gears here from our the case studies and just give some of you all a preview of some new developments that Freeslate's working on before we move on to kind of concluding marks and some questions. Some of you all may have been familiar with some of the things I talked about previously, and some of you may not. But we are continuing to work on. This is an area that we feel has a real need, and we're continuing to work on to develop this for high throughput research. The first is for pH measurements. We've been doing pH measurements in an automated fashion for some time. We've used it for small molecule research and doing pH assessment for some time. And traditionally, we've used a glass electrode, which gives a very good measurement, but the glass electrode tends to be very fragile, and it tends to have a slower response time. So currently, we're working on a new solid state type of pH probe. It's an ISFET-based pH probe. It's an extremely robust solid-state sensor. There's no glass. It has extremely fast equilibration time, so it takes less than 5 seconds to equilibrate, as opposed to the 30-second equilibration time of the glass, which means as you're stepping through a series of an array of samples, you can go through much more quickly. At the prototype stage we've been working on so far, we've demonstrated a 3x increase at least in throughput and a higher measurement repeatability from sample to sample. It has a very high resolution. It's got an integrated temperature sensor, and all these things are an advantage. And finally, it's a sensor that can be stored dry. The glass electrode is a probe that needs to be stored in a storage buffer in a wet condition. Normally, in a manual mode, this is not a big deal. But if you do it in the robot, that means that between every sample, you need to store this thing, you need to wash it. It adds extra steps, and it complicates the process and it slows things down. So as we've been looking at this, the other thing that we're actually doing is to enhance the throughput even more, as we actually are developing an ARM element that is going to have it'll have 4 of these in parallel, a 4x assembly of these probes, one of which could be extendable. So one could do either a single measurement, or you could do 4 at a time of independent measurements to really look at doing early assessment and doing pH measurement, because pH is a critical measurement for early formulation development. Secondly, and relative to the poll we took earlier, is I think there is a strong amount of interest of doing on-deck viscosity and being able to screen for that. One of the big issues that there is for viscosity measurements is that typical and traditional measurements for viscosity tend to use a lot of material. So we've been looking at a system that is based on a capillary viscometry that's loaded with the PDT as a positive displacement tip robotically. So it can pull out a small amount of sample and inject it directly into our on-deck analysis unit, and to be able to capture data. It has a range of from about 1 centipoise, which is about the viscosity of water, up to over 100 centipoise, which pretty much covers the range that one would care about when looking at biological formulations and things that could possibly be injectable. And by the time you get up over 100 centipoise, it probably isn't usable at that particular point. Preliminary data demonstrate a pretty robust approach to a very low-volume, high-throughput viscosity measurement, and we're excited about this particular approach. Finally, we're actually doing a fair amount of work on looking at the automated detection of visible particles. I mentioned before and showed some pictures of the automated visual inspection. Automated particle inspection process this process we're using is based on a fairly well-defined and documented approach where you'd have a sample in a vial. The vial itself is spun, and you stop spinning it, and you wait a certain amount of time, and you inspect. You use a camera to actually inspect the camera, taking a series of images or a video or some such thing, and you're looking for particles that are moving and using image analysis to detect what kind of particles could be there. We've got some target specifications of how many vials we're trying to do and what kind of throughput we're looking to achieve. And we're setting this up to be able to handle a variety of different types of container enclosures and different types of vial sizes. To date, we've been able to look at essentially, I think we're pretty confident we can cover visual particles. And I'm going to show a slide on the next slide showing imaging of 90-micron polystyrene spheres. So I'm showing here what can be imaged. This is imaging a sample. This is in a simulation vial. And you're actually looking at a collection of 90-micron polystyrene spheres in water. And then what one would do is there's a image analysis program that runs in the background of capturing those. It can do various things to subtract off the background and capture those and do blob analysis to actually analyze the image itself and be able to pick out particles. And this can become important if you're looking at trying to compare different types of formulations and try to assess are particles appearing? Are particles appearing under various stress conditions? How robust is my formulation? Do I need to make adjustments to my formulation to improve robustness? Great. Well, I think I've got a few kind of closing remarks before we come to questions. I wanted to wrap up talking a little about working with Freeslate. Generally speaking, what we do and what Freeslate does is we're focused on developing these types of workflows, developing high-throughput approaches, and being able to deploy them out to our customers and our partners' laboratories to enable them and their researchers to do these approaches. So our primary thing that we do is comprehensive workflow delivery, and it's really more than just delivering a piece of automation equipment. We develop, we validate, we deploy, and we support these workflows. And the workflows is really from end to end, including all the software and everything about capturing the data. We have a number of standard workflows, and we also do a fair bit of tailoring of the workflows to customize to meet customers' needs as well. We do have labs here in Sunnyvale, and so we are able to run proof of concept studies to actually show and demonstrate chemistries for customers to make sure, to ensure that the workflow works on the chemistry that's needed and help choose what type of configuration could be the right configuration for the lab. And we also do technology development, both for our own development of knowing where needs are in the marketplace, as well as in partnership with some of our customers as well, who may have specific needs for particular unit operations or particular types of functionality that doesn't quite yet exist. We've done this over a number of years. Freeslate itself is a young company. I think the name is just over about 14 months old. But we've operated as part of an operating division for about, as part of a company that was operating for about 16 years. And over that time, we have a track record of developing things to companies like Merck and Eli Lilly, and even Dow, Pacific Northwest National Labs. These are companies that have talked about the workflows we've brought in and talked about the large impact and benefits we've had with the workflows we've done over time. And so I think with that, I'd like to conclude just by saying that, I think that high-throughput research and high-throughput solutions can really impact the formulation challenges of today. I think being able to fully automate and integrate systems for biophysical characterization and stability studies can be very important. It allows you to be consistent and accurate with your performance of generating and exploring a broad space of formulation space. We've really looked to design something that can integrate a number of different third-party equipment and be flexible, both for initial setup and so it can evolve with changing needs. Software itself seeks to be intuitive software to help advance decision-making. Not only can it integrate a number of different analytical stations, but you can view from different types of studies, tie things together, and you can easily share information across an organization. And all that really seeks to be able to explore a broad range of formulation space to enable knowledge-based decision-making and to really enable risk management early in development. So I think with that, I will thank you all for your time, and I appreciate you hanging with me. And I think we're going to have a session on questions at this particular point.
Fantastic. Okay. Thank you very much, Eric. Okay, ladies and gentlemen, that's the end of the first part of today's session, which is obviously the formal presentation. We've had a number of questions come from you during today's session, so thank you so much for submitting them. We've responded to one of two of you via the messaging system, and we've still got a number to actually get through in the Q&A, so we will answer as many as we can. If you have more questions, please submit them now. If we don't get to the question during the live session, then I'll share them with Eric after the session, and I'm sure he'll come back to you direct. Just to make you all aware, ladies and gentlemen, a copy of the slide deck will be available on our website within around about 24 hours of today's session. We'll notify you in email once that is available. You'll simply have to pop to our site and click download, and in the same location you'll find a copy of the recording of today's session. So we've got a number of questions that are already prioritized. I think, Eric, you're just having a look at them there as well. So once you're ready, please let me know, and we'll start working our way through these questions. I don't know if there's any there as well. You very quickly want to prioritize yourself as well.
Absolutely. So, well, why don't I start in and take a look at some of the questions that may be kind of common. So we had a question that came in that says, these seem like fairly involved experiments. How simple is experimental design, and how could the data then be used to drive decision making? So I mentioned about the software we had earlier. The software itself in the experimental design is fairly intuitive. The software package allows for a fair bit of complexity, both in terms of thinking about composition space as well as parameter space. But it's set up to be very graphical in nature. It can, and set up to be very quickly be able to either add things in gradients or add things in equations. And I would say most researchers find it very intuitive and learn very quickly. The graphical nature also makes it fairly easy to share your design space and what you're trying to do with your team members as well. That data then gets saved to a database and published as a recipe that has a identification number with it, which means that's exactly what carries forward to the sample preparation robot as well as to any of the analytics as well. And it really was what ties everything together.
Okay.
Yes, go ahead.
I was going to say, we've got a few here, then, I don't know if you see in the list here, that are kind of coming a little bit earlier on in the session, that have been kind of prioritized by the team in the back. Should we work our way through them from the bottom as well, Eric? I think it starts with would the viscometer, visual inspection station, and pH meter, that one? Should we work from that one?
Yes. So the question is, would the viscometer, visual inspection station, and pH meter be available to be incorporated into a Type 1 benchtop robot? So I believe the answer is yes, it could. It would depend on the robot itself is a 1.5 meter robot, which enables a number of different possible of elements to be on the deck. What it may do is it may limit the types of other types of mixing stations and how many other types of samples and preps and different source materials could be on the deck. There are some trade-offs in terms of just the geometry and the space that are involved. But certainly that type of thing could be included on that Type 1 type of a standalone system. And it's one of those types of things that one could, over time, add on either other types of virtual instruments or other types of things over time as well if needs grew and changed. The second question here is, do you have a list of machines of software packages that Freeslate can be integrated with? The answer is that we do. We have a history of doing things, and it obviously depends on somewhat on the software version and what instrument, specific instrument model we're talking about. We've done a number. We'd be happy to follow up at some point afterwards and talk specifically about that. We've taken an approach generally to be fairly, or attempted to be instrument agnostic, so that we know that analytical researchers use a variety of different types of equipment. Some people, for HPLC, for instance, since that's an example that many of you all used, some people use ChemStation and Agilent instruments. Some people use Waters and Empower types of systems. Our integration is common. We can talk with both those and other types of chromatography instruments, and all gets captured back to the database in the same way. Yes, go ahead.
We've got another one here. I think I've been asked to prioritize. You mentioned virtual integration when describing the HPLC. Could you explain in more detail what is meant by virtual integration?
Absolutely. So what we found over time is that we talk about integrating workflows quite a bit, and it can mean different things to different people. Integration for us always means it's integrated with the software. The software itself is connected, and so that wherever you are, when you walk up with when you hand that instrument a piece of an array of samples, it reads a barcode or you type in an identifying number. It knows where those samples came from. It knows what is the history of those samples. It can build out a sequence table in an HPLC, and it can store that data relative to that sample in the database. Now, it may or may not be physically integrated to the robot. So a virtual integration is our way of talking about a system that is integrated to the software, but isn't connected to anything else hardware-wise. So what it means is an actual researcher, a person, would carry a plate of samples up to that instrument, the chromatography instrument. You'd scan in the barcode, you'd hand it off, and you'd start the instrument, and the data would all be collected and saved to the database. That instrument might be in the same lab as the robot that generated the samples. It could be down the hall. It doesn't really matter where the instrument is if it's virtually integrated. A physically integrated system would be something that is actually connected, where the robot itself picks up a set of samples, hands it off, and delivers it directly to the sample itself. Yeah.
Fantastic. We've got another really good one here as well. I've been asked to go to Eric. Would you be able to implement existing equipment, i.e. DSC, HPLC, into your robot, or does it need to be purchased new?
So we've done both those things. It very much depends on the make and model of the instrument itself, and so it's one of the things that we should probably follow up with offline. I will say that past experience has shown that in some cases it may make sense to some analytical instruments. It is additional capital. Although if you're talking about suddenly going from doing manual experiments to doing high throughput experiments, it may be that the capacity is such that you have enough experiments that getting an additional unit, analytical unit, makes sense. But it isn't always required, and oftentimes we do integrations. Virtual integrations are quite easy to do, and we often wrap virtual or software virtually around instruments that are in the customer's lab already. Physical integrations, the only limitation there is that if we're doing a physical integration, we're joining things physically with our robot. The limitation is just the instrument has to come out to our lab so we can do the actual physical integration. We can test things before shipping it back to the customer's lab as well.
Fantastic. And just referring, I think, back to a previous answer your answer to the previous question there, Eric. It says, is this a mixing station, or could it be used for buffer exchange?
Right. So the answer is buffer exchange. Certainly, if you're talking about making a set of samples, screening it early on, the ability of formulating early samples can be very important. One of the other developments that we're actually working on is looking at some different ways of facilitating high throughput buffer exchange. I think it depends on what one needs to do and where one is. I think that if there's interest there, I think we'd be happy to follow up with folks about what our thoughts are on that offline, because I think it bears further discussion with interested parties.
Fantastic. Okay, well, we're probably a couple of minutes from the hour now, Eric. So I don't know if there's any in particular you can see in that. And ladies and gentlemen, I will tell you we've got an awfully long list of questions here. So sorry if we're not going to get to every single one of them. But is there any that kind of that stand out to you before we go?
Well, so a couple here. So a couple people asked about the volumes, and could I comment on the volumes used for biological samples? And I guess I didn't mention that going through. We have worked at small volumes before. We've done things down at about, it'd be typical for us to do samples at less than 500 microliters. Several of my examples I talked about doing at 250 microliters per sample. And we have done samples at like 125 microliters or so. At a very low sample, you start getting limitations on, it depends what analytics you're going to use. Things like dynamic light scattering and plate readers don't use much sample, and you can get away with very small amounts of sample. Some types of analytics, you need more sample afterwards, and so it very much depends on what you're trying to measure and what you're trying to assess. But you take a look at that overall, and you design your experiments accordingly. Anything else here? I guess the last one I can mention is that a couple people asked about the visual inspection station, how it works. Is it a tool that is part of the deck of the robot? And the answer is that it is. So you can picture a station on the deck that has a camera sitting in it. The robot itself has a vial moving arm. So it comes out, it plucks out a vial out of your array. It moves that array into an on-deck element. It closes up a station. So it's basically a light enclosure. It has controlled lighting inside with a digital camera, so you can control the lighting and you can control things. And it captures an image, or it captures a series of images stored in the database. The robot then picks up the vial, puts it back, and grabs the next sample.
Fantastic. Just to let you know, we're now about 1 minute from the hour mark, so we've probably not got much more time to get through many of the other questions. I think it may be better, would you like to make a closing statement just to the audience before we tie up today's session?
Certainly. So my contact information is here on this final slide. I'd encourage people who would like to have a follow-up. We'd be happy to follow up offline. I know there were probably questions I wasn't able to get to, and would be happy to do that. I want to thank everybody for their time today and their attention here, either in late morning or afternoon or wherever you happen to be. Actually, this is an area that I'm excited and passionate about, and I like to talk to other people who also are very interested in this particular space. Thank you very much for your time today, and I look forward to talking with more of you as we move forward.
Fantastic. Well, thank you very much, Eric, and thank you to everybody at Freeslate who've been working very hard in the background to make sure we have got through as many of those questions as possible. And ladies and gentlemen, if we haven't managed to get to your question, we do apologise. I will share them with Eric and the team afterwards and leave them to follow up with you. As mentioned earlier, the recording for today's session and the slide deck will be available. We will notify you in an email within the next kind of 24, 48 hours. So I'd just like to thank you all for your participation and your attendance, and I wish you all a very good day.